Hi, I'm Mike Blossberg, the Biology Education Manager here at PASCO, and today we're going to do a quick lab on photosynthesis using the wireless CO2 sensor. To get started, I'll power on the sensor and launch SparkView and open the Bluetooth menu. The red LED next to the Bluetooth icon indicates the sensor is ready to pair, so I'll find the corresponding 6-digit ID number in the Bluetooth menu. Once I've connected to the sensor, it'll move up to connected devices in the software, and the Bluetooth LED will begin flashing green. I'll click Done to close the menu. Today we're going to be using spinach as our model organism to study photosynthesis, but any dark green soft cuticle leaf should work well. We'll place just a couple of leaves into the sample bottle that comes with the sensor. Because the sensor is so sensitive, 1 or 2 leaves should do the trick. It's important not to have any leaves overlapping so that they're shaded from our light source. We're going to look at this closed system in both light and dark conditions to see how the CO2 concentration changes. Now that I have my leaves in place, I'll insert the sensor and place it under our light source. I recommend using an LED or a fluorescent bulb. If you have an incandescent bulb, you may want to set up a heat sink using a beaker of water between the light source and the sensor to avoid overheating the system. In SparkView, I'll select CO2 concentration, which will build a graph display of CO2 versus time, and we'll begin data collection for about 5 minutes or until the CO2 concentration has changed by at least 100 parts per million. Okay, it's been about 5 minutes. I'm going to go ahead and stop data collection, and we'll continue with the second run before doing our analysis. For this run, I'm going to wrap the sample bottle in tin foil to block out all external light. You can leave the sensor and the sample bottle in place. And we want to wait about 1 minute before beginning data collection for another 5-minute run. All right, I'm going to go ahead and start data collection, and we'll proceed for 5 more minutes. Okay, it's been about 5 minutes. I'm going to go ahead and stop data collection, and we'll do our analysis. First, I'll rescale the data. Next, let's look at some of the statistics for each run. I'll select run 1. Let's look at our minimum and maximum carbon dioxide concentrations. As you can see, the carbon dioxide level decreased by approximately 200 parts per million, indicating a net consumption of CO2 as the plants were exposed to a light source and conducting photosynthesis more than they were conducting cell respiration. For the second run, we perform the same analysis. We can see that the carbon dioxide level increased by almost 500 parts per million, indicating that respiration had become the dominant metabolic pathway. This is a quick and easy setup you can use to introduce your students to photosynthesis and get them started doing inquiry labs. They can change the light color, light intensity, plant species, temperature, and more to investigate photosynthesis. If you'd like more information on this or other wireless products, please visit pasco.com/wireless. Thanks for watching.